We're starting chapter 5 this week, and with chapter 5, we're going to take a look at the eukaryotes. So recall from chapter 1, we have two types of cells on Earth, really two divisions of living organisms, and these are the eukaryotes and the prokaryotes. Prokaryotes, if you'll recall, are the bacteria and the archaea, described somewhat as simple in comparison to the eukaryotic organisms. Eukaryotes include things we're typically more familiar with. These include all the animals, these include all the plants, these include things like mushrooms and fungi. Also the single-celled eukaryotes, which is the protista kingdom, things like algae and protozoa. So while there's a lot of differences within the eukaryotic group, plants and animals are very different, obviously. What we're looking at today, in part 1 especially, are the characteristics that you find in common amongst these organisms. So really we're looking at the cell characteristics primarily. These include things like how the DNA is organized. This includes topics on different cell organelles, and a little bit about how some of those function and contribute to the overall functionality of the cell as a whole. In part 2, we'll take a look at some different examples of eukaryotic organisms. We'll take a few notes on how they might contribute towards health and disease, and provide some examples of different types of eukaryotes that would specifically be studied in microbiology. So we're not going to look at all the different organisms. That would take a whole class and then some, but just a few examples of how eukaryotic organisms actually fit into microbiology. Okay, so we'll go ahead and start with part 1. And in part 1, we're going to take a look at things like the cell organelles. We're going to take a look at the DNA. We're going to take a look at the cell membrane. We're going to take a look at also some of the levels of organization towards the very end. How eukaryotic cells, for example, group together to form tissues, and how that then can lead to things like organs and organ systems. Okay, so let's go ahead and start with the very first section of part 1. Okay, so we're going to start with this a little bit overview or review from chapter 1. So in chapter 1, I kind of outlined some basic differences between eukaryotes and prokaryotes. So here's the definition of a eukaryotic cell. This is defined by our textbook: a cell that differs from a prokaryotic cell chiefly by having a nuclear membrane, a well-defined nucleus, membrane-bound subcellular organelles, and mitotic cell division. So that's a very straightforward, or excuse me, a very elaborate definition rather, from our textbook. Really what I want you to focus on, I'm not going to make you memorize that definition or recite that from memory, don't worry about that. Focus more on what you see here, the primary differences between prokaryotes and eukaryotes. And while there are many differences, these are some of the most substantial. So the first one is that their DNA is surrounded by a double membrane. That means that they have what's called a true nucleus, which basically just means DNA surrounded by a membrane. And they contain cellular organelles. And what cellular organelles are units within the cell that operate independently of each other. So I'll draw a figure here in a couple slides and give you a better idea what I mean by that. So really, to summarize, these are compartments, to be compartments within the cell that each take on their own function. Okay. Eukaryotes, here's a review of what types of organisms those are. I mentioned that they come in both single and multicellular forms. So if you see a multicellular organism, this is only a eukaryote, or rather, excuse me, only organisms large enough to see with the naked eye are always eukaryotic. If it's a microscopic organism, it could be either a eukaryote or a prokaryote. So large, multicellular, macroscopic organisms, these are all eukaryotic. Microscopic can be both prokaryotic and or eukaryotic, depending on what you're looking at. So recall that kingdom Protista, one of the 4 major kingdoms here, these include all of the single-celled eukaryotic organisms. And just a couple quick differences here between the two before we go into more detail. Typically, a eukaryotic cell is about 10 times larger than a prokaryotic cell. That's an average, so there's exceptions. And when we talk about the complexity and I said eukaryotic cells are more complex than prokaryotic cells. Some examples of that include the cell cytoskeleton. We'll talk about that later in the chapter. Advanced motility features. Both have like a flagella, for example, but a eukaryotic flagella has a higher level of structural functionality to it, so it's much more elaborate, much more detailed in its design, so to speak. And then I already mentioned the membrane-bound organelles, kind of like units within the cell. Okay, so next we're going to do a quick stick figure drawing of a comparison between a prokaryotic and a eukaryotic cell. So what you want to focus on are the questions here, and I'll go through each of these, and I'm going to do it here on my paint document. So starting out, we're going to make two cells, just represented by these boxes, and one's going to be about 10 times larger than the other. That's not in a perfect ratio. It's not as important that you draw them at the right scale as just understand that the eukaryote is much bigger than the prokaryote. Okay? And for later purposes, recognize that the ratio should be approximately 10 to 1 in terms of size. So the eukaryote is on average 10 times larger than the prokaryote, even though that drawing there isn't really a 10 to 1 ratio. Okay, so there's our first part. How much larger should it look? Next up, we're going to do internal structures that are found in both cell types, and then we're going to look at structures only found in the eukaryotes. So, create a little more space here, and right down the middle, I'm actually going to do a comparison showing the types of structures that you would find in both a prokaryote and a eukaryote. Okay, so I'm just going to put common to both here in the middle. Okay, so what I'm going to put here, common to both, these are generally structures that you would find in virtually any living organism. So, these include things like a cell membrane. Talk about that in the next here, coming up in chapter 5, part 1 here. But a cell membrane is common to all living things. It's a characteristic of life in itself. Common to both, we would also have DNA. We would also have a cell cytoplasm, and we would have ribosomes. Now, I talked a little bit about each one of these in chapter 4 when I talked about prokaryotes. So let's just real quick. A cell membrane is basically a lipid bilayer around each cell. Now, we talked about that in the context of in chapter 4 with the cell envelope. So the cell envelope has the membrane, cell wall, and sometimes the outer membrane. Okay, so in this context, we're really just talking about the cell membrane, and it's important to recognize once again that all living things have one of these. Now, some living organisms, like certain bacteria, they have a cell wall in addition to that. They might have an outer membrane in addition to that. So there's many other layers that they could have in addition, but they'll always have, as a minimum requirement, this kind of basic lipid layer around the cell called the cell membrane. And we'll get into more detail on what that looks like here shortly. DNA, just going to use the letters DNA here, is always found somewhere inside of a cell, and it's another key requirement of all life forms. Every living thing has DNA, and I'll talk about the difference between the two when I get to the eukaryotes. Cytoplasm is just the liquid inside of the cell. So I'm just kind of pointing to nothing here in the cell. Just recognize it's the fluid inside of the cell. It's called the cytoplasm. And then ribosomes are these little, very tiny structures that in a prokaryote often are just somewhat floating around in the cell. So here I'm kind of just drawing these, just kind of floating around here. They can just be anywhere in this vicinity. And you'll find the same structures in the eukaryotic cell, but as we'll get to them here in a minute, we'll see that they're a little more structured. But I'll go ahead and put them here. They're typically centered near the DNA, and I'll talk about a little more in detail about what else goes with the ribosomes here. So these are structures, once again, you find in all living things. They'll be a little different in terms of how big they are, how much there is, where exactly it's found, but a membrane, DNA, cytoplasm, and ribosomes. Now let's take a look at the eukaryotes, and we're going to talk about the structures that would only be found in the eukaryotes. So we're looking here, what internal structures are found only in eukaryotes? So I'm going to put these over here. Unique to eukaryotes. Okay, so we're going to start with the nucleus. Okay, and earlier I said that a nucleus, or excuse me, I didn't use the word nucleus. I said that here the DNA of a eukaryote is surrounded by a double membrane. So the DNA, being central to a cell in all living organisms, but especially in a eukaryote, has its own separate boundary around it. So here's the DNA. Instead of it just being somewhat off by itself, it actually has its own double membrane around it. So we'll draw this double layered membrane here, and we call that the nucleus. Sometimes it's called a true nucleus, okay, but for short we just call it the nucleus. Compare that with a prokaryote. They have DNA, it's actually the same stuff really, but there's no separate compartment around it, there's no separate membrane around it. So there's a distinct difference between these two organisms in that the DNA is separated by a membrane and not separated by a membrane. So that's the nucleus. Next, we'll talk about how these ribosomes I mentioned are organized. And there's a structure here called the endoplasmic reticulum, and there's actually two types. One type is called the rough endoplasmic reticulum, and that can be abbreviated as RER for rough endoplasmic reticulum. And then you've also got smooth endoplasmic reticulum, abbreviated SER. So rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, now the rough endoplasmic reticulum, we'll talk about that first. It's considered rough because it is in association with the ribosomes that I already kind of drew here. And the ribosomes are somewhat studded along with this endoplasmic reticulum. And the combination of the endoplasmic reticulum itself plus these ribosomes embedded within it give it what was said to be a rough appearance. Okay, so that's where the rough comes in. What it really is, though, is it's another layer of membrane that folds outward from the nuclear envelope, or from the nuclear membrane here. So this is the nucleus, this is a separate membrane here. And the endoplasmic reticulum folds out from that. And we have a series of kind of flattened sacs that fold out from the membrane here. And once again, within them, we have these ribosomes that give it a studded, kind of rough appearance. Now we'll talk in detail, a little more detail about what the RER does, but for now I'm just kind of drawing it and where it's located. Smooth endoplasmic reticulum is similar, but it's a little different, and it tends to branch out near the ends of the endoplasmic reticulum, sometimes off to the sides. It doesn't always look like this, by the way, but it tends to kind of branch out from the RER in different areas here, and it's considered smooth because it lacks ribosomes. So you see the distinct texturing here that I've drawn with the rough, and then the smooth is characterized by a lack of those ribosomes, giving it a smooth texture. This arbitrary name someone came up with. There are different functions for each, and we'll talk about those shortly. But for now, recognize where they're located and the difference between them being ribosomes and a lack of ribosomes. So that's the endoplasmic reticulum, the two types there. Next, I'm going to talk about the Golgi apparatus, sometimes called the Golgi body. Golgi body, or sometimes Golgi bodies. Okay, I usually use the word Golgi apparatus, but you might see it in other forms there. Go ahead and just keep it to the Golgi apparatus here. Golgi apparatus looks a little bit similar with my real simple stick figure drawing anyway. And it's another series of flattened membrane sacs, often occur just adjacent to the endoplasmic reticulum. So I'm going to draw that somewhat just off to the side over here. It's usually a certain layer of these membrane sacs that kind of stack on top of each other like this. Now the Golgi apparatus has its own function, but I'm going to draw a little arrow here in conjunction with the endoplasmic reticulum to indicate that these two structures work together. So the RER and the SER, here's the RER, here's the SER, these work in conjunction with the Golgi apparatus, the Golgi bodies. Okay, we'll talk about how that works shortly, but for now, just recognize that's what the arrows are representing. So all of these so far are really separate membranes within the cell. So you've got the DNA membrane around the DNA, and then these here are examples of organelles. So that's an organelle. Rough endoplasmic reticulum, that's an organelle. Smooth endoplasmic reticulum, that's an organelle. Golgi apparatus, that's an organelle. Let's talk about another organelle called the mitochondria. Mitochondria, often referred to as the powerhouse of the cell. This can be actually located almost anywhere in the cell, but I'm going to draw it down here. Another one that's got its own double membrane around it. So this one's got two membranes. It has kind of this folded internal structure that might look like this. Not the best drawing there. Kind of has this shape like that to it. Fill this in for contrast. Mitochondria is often referred to as the powerhouse of the cell, and it makes energy. That's what it does. Okay, we'll talk a little bit about that as well. And I don't always draw these on here, but I'll go ahead and add this one. You have another common organelle called a lysosome. Lysosome or lysosomes, you sometimes have multiples. Once again, these can be located kind of anywhere. They're kind of these circular vesicles. Sometimes you'll have multiples. And they'll often contain chemicals within them that help serve their function, which is digestion. So draw little specks in there. Those represent little metabolic enzymes. And most cells contain these. So those are fairly common. Okay, now this doesn't include everything. Plant cells will have additional organelles like a chloroplast and even a plant vacuole. They'll have cell walls and additional structures. So what I've really drawn here is a basic comparison with the structures that you typically find in all eukaryotic cells and how that would compare to the structures that you would typically find in a prokaryote. But if you look here, just as a quick side-by-side comparison, the prokaryote has very little internal complexity going on. It's got some of the core basic elements, but inside there's really not a lot of detail to how they're structured. In a eukaryote, we see that there's a much higher level of organization. The structures kind of all separate within the cell. And the way to think about this, I like to use this analogy anyway, is you can think of a prokaryote as almost a single room apartment, or almost like a log cabin, like a studio apartment. If you ever lived in a studio apartment, you know what I'm talking about. Just basically kind of one room where you've got almost everything happening. A eukaryote in this analogy is more like a traditional multi-room house, where you've got the house itself, which would be the cell, but within the house you've got separate compartments. So you've got the nucleus, which might be kind of like your master bedroom, and you've got the endoplasmic reticulum, which might be like the living room, and then you've got the kitchen, and you've got the bathrooms, and you've got the study room, etc. Right? So each part of the house kind of has its own function. They all work together as a unit, but each one kind of has its own thing going on. And this is true of the eukaryotic cell. What this allows is for specialized processes to take place in one part of the cell without affecting the rest of the cell and without interfering with something else that might be happening. So for example, these lysosomes have digestive enzymes that could potentially affect the rest of the cell if they were to leak out of here. So their effect can take place within the lysosome, independent of the rest of the cell. So these potentially damaging enzymes don't affect the DNA because everything's separated. In a prokaryote, they don't have that luxury. As a result, what can happen in the cell is somewhat limited, because if anything too radical takes place, it might affect the DNA in a negative kind of way. All right? So let's go ahead and move on to the next part of our chapter here. Backed up here. So what I'm going to do next, I'm going to start with the cell membrane, and then we're just going to go through each one of these different examples here and talk about them in a little more detail. Okay, so we mentioned the cell membrane. Said all living organisms have one. What a cell membrane really is, though, is it's a collection of a certain kind of molecule called a phospholipid. This is at least the primary molecule found in the membrane. So here's a picture showing just a membrane around the cell. This is a eukaryotic cell with all the different things happening in here. Sorry, I got interrupted there. Forgot what I was saying exactly. The membrane is made up of phospholipids, which are a certain type of molecule that make up the base of the membrane here. So this is a cell showing the membrane surrounding the cell. This is a cross section showing the inside. This is a eukaryotic cell here. So phospholipids are unique in that they have properties to them that consist of two distinct regions. So they have what's called a head and tails, typically two tails in a phospholipid. And the head is said to be polar, which means it has a charge to it. And the tail is said to be nonpolar, meaning that it does not have a charge. So it's a neutral region. So a neutral and then a charged region of each molecule. So here's a few diagrams showing what the membrane would look like. So I mentioned we've got the phospholipids with a polar head and a nonpolar tail. One of the implications of being polar is that it makes it dissolvable in water, and we call that hydrophilic, meaning water-loving. So this basically just means that it's not repelled by water and that it's actually able to be dissolved in water. So anything that actually dissolves in water is hydrophilic and somewhat polar. So those properties all go together. So the heads are hydrophilic, and that means water can move right up and immerse around the heads of the cell. And so the same is true on the outside of the cell and on the inside, where we have the heads in both directions. So we have two layers of phospholipids with the heads pointing outward from each section. The tails then, which are hydrophobic, nonpolar, they point towards the middle, like you see here. And what that does is it creates a distinct zone in between these here that is hydrophobic, or water-fearing, or water-repelling is another way to say it. So in a short context, what it does is it creates a zone in between these two layers of phospholipids where water does not go, and creates a distinct boundary between the inside of the cell and the outside of the cell. And all cells are made up of a layer that looks very much like this, almost nearly identical in every cell, with phospholipids pointed in each direction, hydrophobic middle here, and hydrophilic on the inside and the outside here. Okay, so this is a real basic figure. This here is a more three-dimensional figure showing kind of the same basic structure. And then this figure up here is showing the phospholipids plus what's not shown either in these other two figures, which are some of the proteins and other molecules that are embedded within them. So I'll talk about those here just shortly. This is just a little bit of a recap of what I mentioned there. Cell membranes orientated with the polar heads facing out and the nonpolar tails facing the middle. So that creates a hydrophilic property on the inside and the outside of the cell, and a hydrophobic property on the inside. So what that does, and so just to clarify, since all cells, back up here, all cells need water. This allows water to interact on the inside, water to interact on the outside, but a distinct boundary between these two regions to keep the cell somewhat of an isolated unit between the outside and the inside. So it just kind of creates like a wall. It's almost like every cell is like a little village with a distinct wall around it, like a castle with a wall. And the wall is like the membrane. In the middle of the wall is this kind of this no-go zone where you have that boundary between the inside and the outside. Okay, so I mentioned the proteins in the membrane. Let me back up just real quick here. Every membrane is not just made of these phospholipids. The phospholipids are like the base of the membrane, but what you'll have added to it are a variety of different proteins, and they take on a variety of different functions. So you have those that act as a little channel, like a gate, so to speak, a transport protein channel. These allow things to move in and out through this opening here. You have other proteins that act as almost like a wire that can send a signal from one side to the other, almost like a telephone wire, where the signal goes through and is communicated to the other side of the cell. Some proteins act as little ID tags. They stick out. It's almost like a name badge on each cell. And many other functions as well. I'm not going to get into too much detail, but I do have a list of basic functions here. So transportation is like a little channel. Attachment is almost like a little latch on the outside of the membrane, where proteins can kind of allow cells to stick together. I mentioned the wire, almost like a telephone wire for communication and signaling. I mentioned the ID tag for identification. And then I didn't talk about metabolic activity, but that is another action that can take place. Certain enzymatic processes, like a breakdown, can take place within the membrane. Okay, so just be able to list a couple of examples, or at least recognize some basic examples here of the functions that proteins serve in the membrane. And more importantly, just recognize that the membrane really is made up of two key things here: the phospholipids themselves, and that makes the base of the membrane, but then the proteins, which make up the key interactive parts of the membrane. I almost like to think of the membrane, the phospholipid of the membrane, I almost like to think of that as like a wall. So imagine a wall in wherever you're at, in a classroom or in your house. And if you look on any wall, every wall's got something on it, at least most do anyway, right? They got clocks, they've got outlets, they might have a bookshelf, they might have a window. Every wall usually has some kind of feature to it, right? Even if it's just a basic outlet, there's something there that is added to the wall to give it more functionality. And that's kind of like the membrane. You got the wall itself plus the windows, you've got the telephone wires, you've got the windows to the outside world. All these things are part of the overall structure. Okay, and then this is just a figure showing some different examples. Transport, enzymatic activity, things are kind of metabolized as they move from one part of the membrane to another. A signal is transduced across the membrane. This is a identification tag. This little green thing acts as kind of like a little ID badge for the cell. Two proteins can allow the cells to join together, almost like Velcro, sticking two cells together. And that can take place in a more elaborate form. Here is what we're talking about with the cytoskeleton. And we'll talk about that more later. So these are just little figures going with those examples there. So next we're going to break down the different cellular organelles that we talked about earlier, give some detail about what each one does. So first of all, an overview about the cellular organelles. I mentioned this already, but these are one of the key features of eukaryotic cells in general. Each is almost like a room of a house with its own unique function. You know, your bathroom is specialized for cleaning and toilet duties, and you would not necessarily want those activities to interact with, say, cooking in your kitchen. They could, but for good reasons you want to keep those things separate, right? So something that's very obvious to our everyday life. In a cell, you have similar scenarios. You have one process that would potentially interfere with another, and as a result, you want to kind of keep the activities separate. And so the organelles provide that function. They kind of separate activities and keep different actions separate within the cell. And in fact, the term organelle actually derives from the term organ. Human organs and animal organs were actually known and well studied prior to the first cells. People were able to see organs long before they were able to actually see cells. So by the time they were actually looking at cells and able to actually see these little organelles, they'd already knew all about the human organs and other animal organs. So they actually realized that these actually look like little organs within the cell. And so that's actually where the term organelle came from. This here is a general eukaryotic cell. It is meant to just point out some of the key features that are common to eukaryotic cells, but probably doesn't really look like a typical, or doesn't really look like any particular eukaryotic cell, rather. So eukaryotic cells can all look very different. They can be different lengths, different shapes, different sizes, and different densities. They can have different material around the outside of them. So each eukaryotic cell can be very different depending on what cell type it is. As a result, we don't really have basic generalizations like cocci and bacilli like we had in Chapter 4 with the prokaryotes. So we're really just going to talk more here about the internal structures, which are more consistent than, say, the shapes, the sizes, and the arrangements, which are highly variable and really specific to each group of organisms. So we're going to focus on the internal content here. So we mentioned the nucleus, the most prominent organelle in the eukaryotic cell. The nucleus is where the DNA is housed, quote unquote, houses the cell's genetic material. And so what I want you to recognize is that the nucleus can be kind of summarized as having two key features. And those are to protect the DNA and to help regulate the DNA. Here it's called the genetic material, but we're talking about the DNA. So protect and regulate the genetic material. So protection just means from chemicals within the cell, also from potential radiating material outside the cell, such as UV light. UV light comes down from the sun every day. It can hit our body, and over time it can cause small amounts of damage to the DNA. Having a nucleus, however, gives us an extra layer of protection from some of the sun's damaging rays, like UV rays. UV rays can still cause damage, but it's an extra layer that helps minimize or mitigate that sort of damage. And the regulation, you can think of the nucleus as almost like a vault in a bank. It's kind of how I like to think of it. Very, very important, valuable information within the vault of every cell, kind of like the vault of a bank. And you don't want just anything moving in and out of that vault if you're a bank owner or a bank manager. So the vault is highly regulated. Only certain things go in, only certain things go out, and everything that does go in and out is monitored and kind of kept in check. And that's really kind of a way to think about the nucleus, is kind of protecting as well as regulating what goes in and out. You know, a vault in a bank also kind of protects the money from potential fires or, you know, any sort of natural disaster. The nucleus itself has three components to it, at least three components. Now, I'm not going to ask you any real in-depth questions on these, so you don't have to worry about this too much. Just recognize the three components. So the first is the envelope itself, and this really is the membrane that is surrounding the DNA. So the envelope really is kind of like the nucleus itself. It's basically that membrane around the DNA. However, within the envelope, you have an extra feature called the pores. Nuclear pores are little openings that occur at different spaces within the nucleus that allow passage to come in and out. Here, that's showing the pores a little better. These are kind of like the gatekeepers of the nucleus. So this is how regulation takes place. These little pores allow some things to come in and out, and other things it prevents from coming in and out. The nucleolus is a little harder to describe, but it is one of the key parts of a nucleus. It is a very dense area of the nucleus, and really what's happening here is a certain type of synthesis, which is the ribosome synthesis. So we talked about ribosomes and the fact that they synthesize protein, but they themselves are actually synthesized within the nucleus here at this site called the nucleolus. Okay, and it gets confusing because you get the nucleus and the nucleolus, and they all sound kind of the same. So that's why I said I'm not going to ask you really in-depth questions, but really just recognize that these are the three components, and just kind of recognize the basics of each one there. So collectively, they make up the nucleus. A quick note on the DNA within, because just to clarify, this doesn't actually show the DNA. So this is kind of like a hollow nucleus. The nucleolus is this dense part in the middle. But what you would have on top of that is all the DNA stacked in there, kind of pressed in there really tightly. It's almost like if you looked at a baseball, if you've ever seen a baseball that's been knocked open, all that stuffing inside a baseball, it's almost like what it would look like. It'd just be chocked full of DNA in there. So the way DNA gets stored in a cell is that it's tightly compacted. So your cells, especially eukaryotic cells, have a really insane amount of DNA. We're talking huge amounts. A cell is very tiny. Each cell is so small we can't see with the naked eye. We already know that. You can fit thousands of cells on the tip of a pin, just to give you a rough idea. If you pulled a cheek sample, like you do with DNA swabbing, if you take a swab and you just gently kind of rub the inside of your cheek, you could easily get thousands upon thousands of cells just with one basic swab. So cells are really tiny. And each one, especially in humans, contains, it's actually over 60, I think it's 6.5 feet, but at least 6 feet of DNA when stretched out end to end. And that's for every single cell, not in total, every single cell. So the way that your body fits all that DNA into such a tiny little unit is it goes through compacting. The DNA is actually wound into tight little balls, almost a lot like how a ball of yarn is kind of wrapped around a spool, or how thread is wrapped around a spool. So the spool is, in this case, the spool is what we call histones. It's just a special kind of protein shown here in this kind of purple. And the DNA wraps around many different units of these histones. The difference here is that you have lots of little spools, and the yarn kind of, in a complex way, wraps around all these different spools together. And then what ends up happening is the spools kind of fit together, and then they start to wrap further and coil tighter and tighter and tighter until the DNA becomes highly condensed. Now that gets complicated. There's different levels of structure that these DNA bands take into, but really just recognize histones are key to the compacting and the folding of DNA, and then recognize that human cells contain about 6 to 6.5 feet of DNA stretched out end to end, and that's how you fit all of that in there. So next we've got the endoplasmic reticulum. I mentioned this in our drawing earlier. Endoplasmic reticulum is actually an outward folding of the nuclear envelope. So here is a hollow cross section of the nucleus, or pores, the envelope here. And if you look real close, the envelope here will actually start to fold outward and differentiate into the endoplasmic reticulum. So the endoplasmic reticulum shown here, parts rough and smooth over here, it's basically these hollow little folded components of membrane that ultimately originate from the nuclear envelope. So at some point in evolution, all of this sort of mutated and evolved from this envelope. At least that's the theory. And in between, you've got these little hollow spaces where substances can kind of move in and out from one part of the membrane to another. But ultimately, it connects to different parts here of the nucleus. So the endoplasmic reticulum is directly connected to the nucleus and is actually an outward folding of the nuclear envelope. Okay, and that's in a general sense kind of what it is and where it's located. Now from there you can look at the two parts I mentioned earlier. You got the rough and the smooth. There's a lot here, so we're going to keep this simple. But the rough endoplasmic reticulum is rough due to the ribosomes. So that's the one thing you definitely want to know. These little ribosomes are actually embedded in the membrane of the rough endoplasmic reticulum, and that's why they call it rough. Now we know ribosomes make protein. So in addition to knowing that it's rough because of those, you want to just know that really in combination, the ribosomes along with the endoplasmic reticulum together specialize in protein synthesis and protein transport. So it's all about proteins here. So they make and help even transport the proteins. Okay, and that's really what the rough endoplasmic reticulum does. The smooth endoplasmic reticulum is smooth due to the lack of ribosomes, and its functions are different and actually much more variable depending on the cell type, depending on the tissue type, could even depend on the stage of development, whether young, middle-aged, or older. Functions can actually change within each cell type. So it's a little harder to generalize here. But some of the things that the SER does do in a more general sense is they help process certain nutrients such as lipids. They're highly efficient at lipid synthesis as well as certain types of nutrient processing. It also can be used to detox certain harmful chemicals. So things like alcohol and drugs can actually be broken down within a cell by the smooth endoplasmic reticulum. And in fact, your liver cells, which are your liver is specialized in detoxifying things like alcohol. Your liver has a specialized type of endoplasmic reticulum that is highly efficient at detoxifying chemicals. Okay. So we're going to move on from that. There's a lot more to be said, but we're going to keep this somewhat simple. Here's a figure just showing, this out of your text, showing the nucleus here and then the different foldings with the endoplasmic reticulum. And then if you were to take a close-up here, you would see here's the membrane and here's the ribosomes kind of embedded in that membrane. And this is a real figure. It's a real high-resolution microscope image showing the nucleus and the endoplasmic reticulum over here. So earlier I drew the Golgi apparatus and I drew an arrow pointing between the Golgi apparatus and the endoplasmic reticulum. And I said the two interact and kind of connect together. So the way the Golgi apparatus works is it takes products that are made by the rough and smooth endoplasmic reticulum and it actually directs those products to where they're supposed to go in the cell. And along the way, it can also modify those products and add some of the finishing touches, so to speak, to some of those products. So for example, a protein might start its synthesis with the ribosomes in the rough endoplasmic reticulum. However, like a lot of products, it comes off the production line in a raw form and it needs some polishing and some finishing touches. So it might get kind of a fresh coat of paint, or it might get polished up a little bit, might get some of the rough edges smoothed off. I'm using an analogy, obviously. And some of that stuff can happen in the Golgi apparatus. So the raw production takes place often in the rough endoplasmic reticulum, but polishing and finishing can kind of take place in the Golgi apparatus. But even more importantly, the Golgi apparatus is often referred to as the UPS, the post office, or the FedEx of the cell. And that's because not only does it put some of the finishing touches, but it actually delivers the product to where it's supposed to go, somewhere either inside the cell or outside of the cell. So when you take a package to the UPS, they receive the package, they put a label on the package, and then they deliver it. And the Golgi apparatus is very similar. It receives a product from the rough endoplasmic reticulum, or this, just to clarify, from the rough or the smooth, from either way. And it actually puts a label on those. It's a biological label, but a little ID tag goes on these products, and those ID tags will actually kind of dictate where they end up. So some of them are directed back into the cell, and some of them are actually directed outside of the cell. Here's a figure showing the endoplasmic reticulum and the Golgi apparatus over here, and how they actually connect are through these little things called vesicles, sometimes called cellular vesicles. Here they're calling them transitional vesicles, and I'm going to talk about those very shortly. Actually, I'll talk about those next. Here's another figure showing the nucleus. Here's the endoplasmic reticulum. Here's some of the products being made inside the endoplasmic reticulum, and what ends up happening is those products are packaged up in these little vesicles that are then transported across this gap in the cell to the Golgi apparatus. So these little vesicles actually carry some of the proteins that are made here and actually deliver them to the Golgi apparatus. And then from there, they fuse with the Golgi apparatus. They migrate through the different layers and are modified and labeled along the way. And as they come out the other end, they are then directed to where they're supposed to go in the cell. So many of these are secreted outside of the cell. So here's a secretion of the product. Some of them are directed back into the cell to be used inside the cell. So the Golgi apparatus has this kind of mysterious way of receiving, labeling, modifying, and then directing the products that are made by the endoplasmic reticulum. And it's easy to not really stop and just appreciate what's really happening here. I was like to just kind of point out this is really amazing stuff here. And this all takes place autonomously, meaning it doesn't appear to be a brain to the cell. I mean, there is kind of the DNA is kind of like the brain, but this all just kind of takes place automatically. This is happening right now in our bodies. Every moment of our life, this has been happening. And it's something that is really complex, and something that you just have to stop and appreciate that's happening. So really you have in your cells really a whole biological factory. And it starts with the nucleus, and the nucleus kind of directs the activity of the protein production, and the proteins get made here by the ribosomes, and then the ribosomes package up some of those products in these little vesicles, and these vesicles kind of float from one side of the cell to the other. And these vesicles, I like to think of them as almost like little bits of Play-Doh. Imagine you got a ball of Play-Doh here and a ball of Play-Doh over here. You can just kind of pinch off a little chunk of that Play-Doh and then go stick it over here and then kind of fuse one piece to another. It's kind of like how these little vesicles work. They kind of pinch off from one side and then fuse with another, and the contents inside of them can get transported like that, like a little capsule. Okay, so that's kind of how the Golgi apparatus works in conjunction with the nucleus and the endoplasmic reticulum. Kind of this one great big long chain of events here, and that's why they refer to this here as nature's assembly line. It's a lot like a factory assembly line. So another note on these vesicles, they call them transitional vesicles here. Here they're referring to them as cellular vesicles, kind of the same thing. Basically, you have the membrane in the cell that, as I mentioned, is kind of like Play-Doh, and it can just kind of pinch off from one section, carrying the contents within it, and then fuse with another section. And so along the way, that process is used to carry the products inside the vesicle. And so this is how the product can be packaged up, pinch off, float around to another area, fuse, and then dump those products into the next layer. So they serve as transport molecules throughout the cell. Real quick, one of the ways that this happens when we secrete things outside of the cell is with our brain activity. Brain cells interact by sending signals back and forth through these little secretion, secretion of neurotransmitters from one cell to the next. Just to give you a quick visual here, here's a nerve cell, and it doesn't show all the Golgi apparatus in here, but the Golgi apparatus will package up these little proteins in these vesicles here, and then when a neural signal comes in, they actually fuse with the outer part of the membrane and then dump those chemicals into this little gap here called the synapse, where they rush across the synapse and then trigger a protein receptor on another neuron, which often leads to another reaction in the next cell. So neurotransmission of brain cells and nerve cells in general is a really good example of where the Golgi apparatus packages up these products and then sends them across from one cell to another. That takes place really fast, very rapidly. Okay, another example, or another organelle here is really a specialized type of cellular vesicle. And this is called the lysosome. I mentioned that one earlier. Lysosomes are a specialized type of vesicle, and they specialize in digestion. So they contain digestive enzymes. And I like to think of the lysosomes as kind of like the stomach of a cell. So our stomach has got all sorts of digestive enzymes, and technically our small intestine as well does a lot of digesting. But you can kind of think of it like that, where within this specialized area, the cell kind of functions to digest things. So part of the digestion of your food takes place within the stomach cavity and the small intestine cavity itself, that we call the lumen. But then parts of the food that aren't fully digested will actually be absorbed by your cells and then fully digested within the cell. So you digest partly outside of the cells within the cavity of your stomach and the small intestine, but then some of those bits of food actually get absorbed into the cell and then fully digested down into their smallest units. So the way the cell does this is it actually swallows little bits of food. It's a process called endocytosis, sometimes called phagocytosis, where the cell kind of just swallows this food particle, wraps it in a special little vesicle, and then the lysosomes will fuse with that vesicle and then dump the digestive enzymes into it to allow them to break down that food. And the food gets broken down, some of the energy gets absorbed as much as possible, and then whatever the cell can't use, the waste is eliminated in different fashions there. So that's what's called a lysosome, digestion. We'll talk more in later chapters about how this can relate. Actually, we talked about that a little bit in chapter 4, about how phagocytosis can be used to break down invading microbes. And we'll talk about that more in chapter 13 later in the semester. But in your white blood cells, this process has actually been specialized to break down microbes, whereas in, say, your stomach cells, it's more specialized to break down food. But it kind of does the same thing, but different depending on the context. Here's just a real quick video showing these lysosomes in action.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
Okay, so kind of mentioned there towards the end that lysosomes are actually used in what they often refer to as cellular housekeeping. So old organelles are broken down, and that allows newer organelles to be regenerated that are more functional. Okay, last two organelles here are grouped together for convenience, and these are referred to as the energy generating organelles. So eukaryotic cells, one of the things that they specialize in, even more specifically than prokaryotes, is that they produce large amounts of energy per cell. So they can really specialize in energy production. So the way that they do this is with their own organelles that generate energy. So mitochondria are really the key organelle here. At least they're the most common, I should say. They're found in virtually every eukaryotic cell. So all eukaryotic cells, with only a few exceptions out there, have a mitochondria. I said earlier, these are often referred to as the powerhouse of the cell. What that means is they generate ATP, usually from glucose, one of your primary carbohydrates, but they can also get it from other sources of nutrients like fats and proteins. So basically, they produce energy in the form of what we call ATP. We'll talk a little bit more about that later, but for the most part, I just want you to recognize energy production, mitochondria. Chloroplast then is similar in that it also produces energy. However, chloroplast is unique only to the plants and the algae. So this is not something that you would find in animal cells or fungi cells or, say, protozoa cells. This is something that you would only find in plants and algae. And the chloroplast allows them to conduct photosynthesis, which is where they take energy from the sun and convert that into glucose. Now, there's a whole lesson on how this works, but in a real brief sense, a plant will utilize both the chloroplast and the mitochondria. So they don't have one or the other, they have both. The chloroplast will actually take the sun's energy and make glucose. So the chloroplast in the photosynthesis process actually makes glucose. And then glucose is like a fuel. And the fuel glucose is then fed into the mitochondria to generate ATP, which becomes the primary source of energy at that point. So the difference between an animal cell and a plant cell or an algae cell is that an animal cell has to get glucose from another source. This goes back to the fact that they are what we often refer to as a consumer. They have to actually eat something in order to get glucose. Plants essentially make their own glucose. So all they have to do is sit out in the sunlight and absorb enough energy from the sun, along with water and other nutrients, to make their own glucose. And then from there, once they make the glucose, they burn it using the mitochondria to generate energy. Whereas again, animals have to get the glucose from another source. So they both use the mitochondria to burn the glucose to make energy, but the chloroplast dictates how they get that glucose, whether they make it themselves or whether they don't make it themselves and therefore have to get it somewhere else. Okay? So those are the energy generating organelles. Next, I want to just mention the ribosomes here. We've already talked about ribosomes. We talked about the fact that they're protein synthesizers. I want to just point out that they function the same in both eukaryotes and prokaryotes. So in other words, they make protein in both types of organisms. These are a core component of all life. A little bit more that we haven't talked about. Ribosomes are made of 2 units, and I like to think of them as almost like a hamburger bun. You get the large bun and the small bun here, and they sandwich together to make the unit that we call the ribosome. So they can actually separate into the large and small subunits. So all organisms have them, eukaryotes and prokaryotes. There is a difference, though. There are 2 different types of ribosomes, and while they do the same thing, they're structurally different. I like to think of the differences between like a Chevy and a Ford. Think of a Chevy and Ford. They're both trucks. They both have 4 wheels and an engine and a truck bed, but the components are just slightly different. And so while they pretty much do the same thing, they more or less look the same way. There are slight differences in the make, the build, and the parts and all of that, right? So there's subtle differences. It's kind of like how the ribosomes are in eukaryotes and prokaryotes. So in eukaryotes, they have what's called an 80S ribosome. And this is kind of like the model number for the ribosome, kind of a random number. There's a logic to it, but for the most part, it's somewhat random. And then prokaryotic ribosomes are said to be 70S. So like Chevy and Ford, you have just really 2 different models here that are just slightly different in their structure. Okay, so recognize that. That'll have implications later. We'll talk a little bit about that in the antibiotic section later in lab. Different drugs, for example, can target one or the other. Certain antibiotics, as an example, target the 70S prokaryotic ribosome, but not the 80S. And as a result, they can actually kill bacterial cells by targeting their ribosomes without affecting the eukaryotes, which have a different ribosome. So there are different implications for having different ribosomes, but otherwise they work the same way. This figure here, by the way, is showing kind of how a ribosome works. It uses messenger RNA along with transfer RNA, and it builds proteins by using individual amino acids. We will talk about that in Chapter 9, though. So that's just a quick diagram of kind of what's happening inside of the ribosome and how they function. Okay, one of the last things I want to talk about are the structures around a eukaryotic cell. So this is kind of similar to like the glycocalyx or the S layer of a prokaryotic cell. However, in eukaryotes, we have a different structure altogether, a different type of structure altogether. It's much more complex, much more variable. We won't have as few groups to put things in here, but talk about it more from a general standpoint. So what we're talking about here is called the eukaryotic cell cytoskeleton. What it really is it's a series of proteins that are structured around the outside of a cell and even inside. So they kind of anchor within and on the outside together and really act as kind of the scaffolding of the cell. So think of a great big building that's got all sorts of internal support beams, and it's got columns, and it's got great big 4x4s in the walls that kind of hold up the structure of the building from the inside out. And if you were to take all the walls down and strip the building down to its studs, and take all the drywall off and everything, you'd see all the structural components of a building that keep it up. A eukaryotic cell is very similar in that it has all sorts of internal support beams. It's got structures around the outside of the cell that give it shape and reinforcement of that shape. And collectively, they're all called the cell cytoskeleton. And here is a figure of a group of cells that have been stained to only show, or primarily show rather, the cell cytoskeleton. So here's a cell, Here's a cell, here's a cell. These are the nucleus, by the way. So each one of these blue dots represents a separate cell. And all these green lines here represent the cytoskeleton that is found within and around the cell. So here's a cell, here's the cytoskeleton within that cell in each one of these here. So collectively, they form kind of this mesh that allows cells to kind of fuse together and reinforces the shape within each cell. So if you want to deduce down the functions, which we've done here, it helps to kind of anchor the organelles. Each organelle is like a room of a house. Well, each room is somewhat structured within the foundation of the house. They've each got their own 2x4s that shape the room and kind of keep it where it's at. This one's a little harder to analogize, but the vesicles within the cell can actually be transported along these cytoskeletal features, and they can also actually allow the cell to change shape and to actually move around. So that's another one that's kind of hard to analogize there, but the whole cell can actually be moved by moving these cytoskeletal features. Now there's a lot here. It's one of those topics where you could really do a whole lecture on just the cytoskeleton. So since we don't want to do that, I'm going to keep it somewhat short here. And I really want to just, in addition to what I already talked about, I just want to mention some of the major components of the cytoskeleton. And I'm not going to ask any real in-depth questions here. So just recognize what they are and a brief note about each one. So you have three types: microfilaments, microtubules, and what are called intermediate filaments. Microfilaments tend to be structured around the cell membrane, towards the outer part of the cell, and these can actually be used for cell movement as well as cell contraction. So they can more or less contract in and out and move the entire cell along with the cell membrane. Microtubules often are found inside the cell and are helpful for maintaining the shape of the cell internally as well as for moving substances inside the cell. Next, we'll talk about flagella and cilia. These are key parts of the flagella and cilia. And then finally, intermediate filaments, also found inside the cell, really help reinforce the structure of the cell and help kind of anchor the organelles inside the cell. So the term intermediate kind of means in the middle of the cell. They help kind of provide the intermediate internal structural reinforcement there. Okay, moving on to the next part, the appendages. We see that eukaryotes have appendages similar to the prokaryotes. So we actually have a little bit of overlap here with the flagella. So we mentioned the prokaryotic flagella. Eukaryotes also have a flagella that's similar. They're similar in the fact that they're both used for movement and they both extend outward from the membrane. They're also similar in how they respond to sensory perception. So something in the environment stimulates the flagella to move. Where they're very different, however, is in the structure. A prokaryotic flagella is relatively simple, whereas a eukaryotic flagella has more layers of complexity to it. And so the flagella is said to have what's called 9+2 arrangement. And I'll give you a figure of what that looks like here in just a second. That arrangement, however, gives it more of a dynamic range of motion, more comparable to an actual tail, and a greater variation in the number of different flagella arrangements. So here's what 9+2 looks like. Here's a cell, and here's a flagella extending out from that. And if you take a cross section, what you actually see is you have a series of these little microtubules I mentioned in the last part there, all joined together. And this is what's said to have 9+2 arrangements. So you actually have 9 pairs of microtubules around the outside of the cell, and you have 2 microtubules in the middle. So you have what's called 9+2 arrangement. And they're all connected by these little fibers. And the way it works is that these fibers can all kind of bend and twist in unison and move the tail back and forth by sliding past one another. So the tail can, for example, pull one of these fibers over here, and that will cause the tail to bend in this direction. Imagine almost like you have little ropes attached to one end. And if you pull the rope this way, the tail moves in that direction. And if you pull the rope in this direction, the tail moves in that direction. Well, it's happening, but just internally. So these little structures allow the proteins to pull down and the tail moves in coordination with the pulling. That may or may not make a lot of sense. I'm not doing a great job of explaining that, but the key part here is recognize the internal structure 9+2 arrangement and the fact that this is much more complex than the prokaryotic flagella that we mentioned earlier. There's a greater number of arrangements. We're not going to get into all the different arrangements. There's a wide variety, so we won't go into all the labeling there. So that's a eukaryotic flagella. Eukaryotic cilia are more unique to just the eukaryotes. Cilia are basically shorter, almost like a stubby flagella. So flagella are like a longer tail, whereas cilia are more like little stubs. They also serve a similar purpose in movement, but they can serve an additional purpose, which is called filter feeding. And these are referred to as oral cilia. I'll give you some figures here in just a second. Let me back up here. This is actually a figure of cilia here. So flagella is more like a tail. Cilia are more just like little stubs, almost like little hairs all around the outside of the cell. And what they have to do is beat in unison, and collectively, all of them moving simultaneously will kind of just push the cell along. I almost like to think of the flagella, if we're thinking about boats in the water, I like to think of the flagella as like a great big motorboat with a great big engine on it, whereas the cilia are like little rowboats. Imagine like a Viking ship or like a rowing team, and you've got 5 or 10 different pairs of oars all moving in unison, and that's kind of how I envision the cilia. So they both move, just different mechanisms of doing it. Now, the cilia can also serve additional function around the mouth or the oral opening of an organism, and they can actually serve in filter feeding. Let me come back to this. Here's a better figure. This here is a eukaryote that's anchored by this structure, and then these little hairs here, these are cilia. And what happens is they move around and trap little food particles in the water, and then allow the food particles to get absorbed into the cell of the organism here. So they act almost like little teeth that can grab onto food particles and allow them to be absorbed. So that's an example of how a filter feeding organism would use them. Here's an example of these are paramecium here, and they have cilia all around the outside of their body, and those are used more for movement. So some organisms use them strictly for movement, some use them strictly for filter feeding, some will use them for both. And these are all just pencil drawings here of different cilia. A couple more things and we'll finish up part 1. Eukaryotic cells can join together to make tissues, and tissues can join together to make organs, and by joining together, eukaryotes can become more complex, working from single-celled up to multi-celled organisms, and then from small multi-celled organisms to large multi-celled organisms. And in a nutshell, the way that starts is it starts in part due to cell connections and mechanisms of joining multiple cells together. So there's 3 basic types of cell connections here. One is called an adhesion junction. These simply connect cells together. So here's a cell next to each other. Adhesion or adhering junctions, as they're called here, simply connect 2 cells together. Gap junctions are little tunnels in between cells, and they allow for signaling molecules to move in between cells so that they can communicate directly and kind of act in unison. Heart cells do this. They kind of beat in unison, and they have little gap junctions that allow signals to move in between all of them simultaneously. Tight junctions are another one, similar to adhering junctions, but their function is more to seal the gaps between cells and prevent fluid from leaking out between them. So these are common in your stomach cells, for example, and they prevent the stomach acid in the lining of your stomach cells from leaking out into the rest of the body. So tight junctions really kind of seal the gaps between cells, so there's a kind of a watertight layer, so to speak, whereas adhering junctions just kind of loosely connect them so that they're still strong but more flexible. So these are kind of 2 different adherent mechanisms, and the gap junctions are more for signaling. So these are ways that cells can kind of group together, as you see here. Cells also are partly grouped together by the cytoskeleton, which we mentioned back here. So some of these cytoskeletal features can actually kind of intertwine together and connect cells that way. So it's really a combination of features that allow cells to group together. But these are in addition to what we've already talked about. So from there, the last thing is just recognize the levels of organization, how a eukaryotic cell would go in a kind of a systematic order from an individual cell to a multicellular organism, potentially. So you start out with an individual cell. Cells will group into tissues, which are a collection of the same kind of cell, which all have the same function, so like muscle tissue. Those cells can then group into an organ, which is a group of tissue, groups of different tissue rather, that all serve a similar function, like heart muscle, like the heart organ, which is made up of different types of muscle, and then multiple organs can function together to make what's called an organ system, like the circulatory system, which has different organs that all work in unison. And then ultimately, multiple organ systems can work in conjunction to make a multicellular organism. Obviously, there's a lot of detail left out there. Most of this is what you learned about anatomy, learn about in anatomy and physiology. But just a quick note on how you actually go from a cell to a multicellular organism through these levels of organization. And once again, that starts in part by connecting cells together and joining them even more so with things like the cytoskeleton to group them. And then once they are grouped together, they can communicate with gap junctions, kind of keep fluid separate with tight junctions, and kind of work together with adhesion junctions. Okay? All right, I'm going to stop there. We'll pick up with part 2 next. In part 2, we'll get into different examples of eukaryotes.